Hello, everyone, and welcome to today's live broadcast, Powering Multi-omic Translational Studies with Mass Cytometry, Characterizing the Molecular Pacemakers of Pregnancy. Presented by Dr. Brice, Gaudilliere, an Assistant Professor, Stanford University School of Medicine, Department of Anesthesiology, and Dr. Nima Aghaeepour, Assistant Professor, Stanford University School of Medicine. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Fluidigm. For more information about our sponsor, please visit fluidigm.com. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen, type your questions into the box, and click Send. We'll answer as many questions as we have time for at the end of the presentation. Also, if you have trouble seeing or hearing this presentation, click on the Support tab found at the top right of the presentation window, or you can report your problem by clicking on the Answer Question box located on the far left of your screen. I would now like to introduce our two presenters, beginning with Dr. Brice Gaudilliere. Born in France, Dr. Gaudilliere studied engineering at Ecole Polytechnique before completing an MD-PhD degree from the Harvard-MIT Health Sciences and Technology program. During his postdoctoral fellowship in Garry Nolan's laboratory, Stanford University, Dr. Gaudilliere developed and standardized a pipeline to implement high-dimensional mass cytometry, cytometry by time-of-flight mass spectrometry, the basis of the CyTOF system in clinical studies. Our second presenter will be Dr. Nima Aghaeepour. His graduate research at the University of British Columbia focused on bioinformatics analysis of single-cell data. Together with colleagues at the National Institute of Health, they established the first objective benchmark for evaluation of algorithms that could automatically identify cell types and eventually correlate them with clinical outcomes, namely the flow cytometry, critical assessment of population identification methods, FlowCap. As a postdoc with Garry Nolan at Stanford, and now an independent faculty member, he has been interested in the intersection of data sciences, immunology, and clinical phenotyping. An alumni of the Stanford Graduate School of Business Ignite program, Nima also regularly consults for a wide range of companies on both technology development and data analysis. Please join me in welcoming Dr. Brice Gaudilliere and Dr. Nima Aghaeepour. I will now turn the presentation over to our speakers. Gentlemen, welcome to both of you.
Hi, thank you very much for the introduction. It's a pleasure to be here and have the opportunity to present our work integrating mass cytometry with other omics platforms to characterize the molecular pacemakers of pregnancy. As you heard in the introduction, Nima and I were both postdocs in Garry Nolan's lab, and we started working on projects together there. And since then, we've been collaborating on multiple research projects. So we thought it'd be fun to give a joint presentation. Sorry. Very generally, our research uses a deep immune profiling of clinical samples with mass cytometry and combines this with high-dimensional modeling approaches to understand how the human immune system adapts to physiological perturbations or physiological stressors. Now, we draw examples of these physiological stressors from clinical studies and clinical scenarios. We're very interested in traumatic injury. Our model is major surgery, surgical trauma, and we're interested here in understanding the human immune response to traumatic injury and identifying immunological events that predict the recovery of patients from pain and disability after surgery. We use a very similar approach in the context of ischemic brain injury or acute stroke. And here we're trying to understand what are the components of the immune response to acute stroke that may be predictive of neurocognitive recovery in patients. But today, the focus of this talk is going to be on our work on pregnancy. 4 years ago, we started a very fascinating collaboration with the March of Dimes Center for Prematurity Research at Stanford, led by Dr. Stevenson. And our goal within this center is to understand the normal immune response to pregnancy with the aim to identify immunological abnormalities that are associated with preterm birth or prematurity. Preterm birth being the #1 cause of neonatal mortality in the world. Now, importantly, all of these projects are done in close collaboration with Martin Engst, who is a professor of anesthesiology here at Stanford. So why pregnancy? Well, pregnancy is a fascinating immunological phenomenon. The maintenance of pregnancy relies on a finely tuned immune balance between immune tolerance to the hemi-allogenic fetus. So for example, effector CD4 T cells and CD8 T cells are excluded from the placenta and the decidua particularly, so that they can't even reach or interact easily with fetal antigens. There's also very important expansion of peripheral Tregs that are fetal antigen specific. That's from the work by Xing Wei that we know is extremely important in the maintenance of pregnancy. Now, these immune tolerance mechanisms have to be balanced with protective immunity against infectious pathogens. And so there's also an exacerbated response from innate immune cells during pregnancy. Now, what's really interesting is that this immune balance lasts exactly 9 months. After 40 weeks, there is perhaps an immunological switch that happens that allows the changes to undergo and lead to the birth of the fetus that you can think about as almost a rejection of the fetus from the uterus. And so clearly the timing of these immunological responses during pregnancy is very important. And this is the major focus of the study that we started. Here, the goal, again, was to understand what is the timing of normal immunological adaptations to a term pregnancy, focusing on maternal immune adaptations. And with the idea that if we understand normal, we can set up the analytical framework to then be able to identify dysfunctional responses or the disrupted timing of these immunological adaptations in pathological pregnancies like preterm birth or preeclampsia. So the first part of this presentation is really going to focus on our analysis of peripheral immune cell responses in maternal blood. And then Nima will tell us about the most recent and exciting work to integrate this analysis with other omics platform that allows us to characterize other pacemakers of pregnancy. So how do we do this? Well, we use mass cytometry, or CyTOF, which, as you know, is a high parameter flow cytometry technology that uses antibodies conjugated to metal isotopes rather than fluorophores. And by leveraging the dynamic range and the lack of spectral overlap of mass spectrometry, the technology allows for the simultaneous measurement of over 50 parameters per single immune cell present in a blood sample. Now, it's important to remember that about half of these parameters in a typical CyTOF panel will be composed of antibodies that recognize phenotypic markers, for example, surface epitopes that allow us to distinguish different immune cells and precisely phenotype these immune cells. And then at the same time, we can use another 20 parameters to assess the activity of intracellular signaling pathways, for example, looking at the phosphorylation form of various signaling proteins, or the intracellular expression of cytokines. And that tells us something about the function of these immune cells. Now, in order to visualize the high parameter mass cytometry data set, there are several algorithms that are applicable, that exist, that all use more or less the same idea to cluster using the phenotypic markers to cluster the different immune cells based on their phenotype, and allows us to build the maps or comprehensive maps of the immune system. So now there's SPADE, there's Citrus, Bisney, Vortex, one of the last clustering algorithm that came out. And here I'm showing you an example from a study that was published a couple of years ago where we used, in collaboration with Matt Spitzer and Federico Gherardini in the Nolan lab, we used a force-directed layout to represent on the same structure the peripheral immune system from the mother and from the fetus, comparing peripheral blood from the mother and umbilical cord from the mother at birth. And what we can do is not only overlay the phenotypic landscape of these two systems, but also the functional landscape. And from this study, we could learn a lot about the similarities and differences between the mature immune system of the mother and the developing immune system of the fetus at birth. We also used a different approach, a similar approach in the context of preterm birth. And that was our first pilot study in this respect, where we compared the baseline immunological state of women that are not pregnant, but at high risk of delivering preterm in their next pregnancy. So we compared women that had a history of term pregnancy with women that had a history of repeated preterm pregnancy. And we used a similar mass cytometry approach to comprehensively characterize immune cell responses in this baseline state. And using citrus, we were able to identify components of this immune system that were associated with this risk of preterm birth. And notably, what we found was that in the myeloid cell compartment, mostly dendritic cells and classical monocytes had an exacerbated response to the TLR4 agonist LPS in women with a history of preterm birth. So now the study that we're going to be focusing on for this talk continues with these application of mass cytometry. But here the question is, from the measurement of peripheral immune cell response in the mother's blood, can we predict the gestational age of pregnancy? So in other words, how accurate can we be in determining the gestational age of pregnancy by looking at immunological adaptations in the maternal blood? So to do this study, we recruited 28 women and separated them in two cohorts, a training cohort where we could build the model that Nima will be talking to us about, and then a validation cohort of which whose samples were kept in the fridge, didn't touch them, and then that was used to prospectively validate the results of the study. Now these women were followed longitudinally during pregnancy and then 6 weeks postpartum, PP means postpartum. And then we collected samples at 3 time points during pregnancy and analyzed them with CyTOF. Importantly, there is a certain variability associated with the time of sampling for each one of these samples. And we use this variability to build a continuous variable onto which we can build a multivariate model that will predict the timing of sampling or the gestational age at the time of sampling. And this is what we call the immune clock model. I want to show 2 slides here that are technical to explain to you in a little bit more details what kind of measurements we're doing. First of all these measurements are done in whole blood samples. And we use the SmartTube system in order to collect and fix whole blood right after phlebotomy, which allows us to really comprehensively look at every immune cell present in these blood samples. And then I want to introduce you to the type of data set that we will be analyzing. So using, I think it's a 43 parameter mass cytometry assay, we're able to precisely characterize about 25 cell types, from your innate immune cells on top to your adaptive immune cells at the bottom. So the yellow dots here are representing the frequency of each one of these cell subsets. But at the same time, we use 10 intracellular markers to look at the functional responses of these immune cells along the line of very canonical and important signaling pathways, the TLR pathways, the NF-kappa-B pathways, and then the JAK-STAT signaling pathways. Now we look at these signaling responses, again, at baseline, in the endogenous way. This is how immune cells are responding to the environment of pregnancy. Those are the purple dots. But we're also looking at them in response to stimulation with receptor-specific ligands. So the blue, the turquoise dots represent the response to LPS. We also measured the response to an interleukin cocktail containing IL-2, 4, and 6, and the response to interferon alpha.
All right, thanks, Brice. We can talk a little bit about how we can take this data set and build those models that predict gestational age. The first thing that we observed in this graph was that the data is actually very modular. This is consistent with what we already know about the immune system, that multiple signaling pathways from multiple cell types correlate closely with each other. And you can see them in forms of these tight correlation modules in this graph. So when building a model that would make predictions, we would have to take that into account. So our multivariate models are fairly simple. We have Y, our vector of gestational age, and we have X, the matrix, that graph that Brice just showed you. And our task is to find a transformation that minimizes the error between predicted gestational age and actual gestational age. That second term to the right of the equation is basically making sure that the model remains constrained, that the model doesn't get too big. We can make the model even more simplified by using a L1 norm instead of a L2 norm. What that does is that it simplifies the model to use only the measurements that are absolutely necessary for making a prediction. That is important because when trying to understand the biology of these models, we don't want to look at a model that's overly complicated for a human to understand. And also when you take these models to a resource poor setting or to a regulatory setting, it would be a lot easier to use a model that has fewer measurements. And finally, we combine these two ideas into a single model that uses both an L1 and an L2 norm to find models that are simple, yet they capture those tightly correlated modules. So we wouldn't pick only one representative from each module. We would capture the entire module if the entire module is correlated with gestational age. Another idea that we incorporated into these models was the idea of incorporating background biological knowledge in the form of Bayesian priors to be able to make more accurate predictions. What we did is that we basically went to the existing literature on immunology and figured out which signaling pathways in which cell types are supposed to respond to which stimulations. This was done entirely without any knowledge of the pregnancy. This is just about our general knowledge of how we think the immune system works. And then we provided this information to the algorithm in the form of Bayesian priors. In a high-dimensional data set like this one, there usually are many models that have a similar predictive power. They are all able to predict, in this case, gestational age with a similar error rate. What this Bayesian prior does is that it encourages the model to find the instances that are more consistent with our current understanding of immunology. We originally did this to make it easier for us to trust the data, to make it easier for us to just understand the biology that we're looking for. But we were pleasantly surprised to see that it actually outperformed a standard machine learning algorithms that didn't have access to this prior knowledge. On the y-axis, we have minus log 10 of p-value for correlation with gestational age. So the larger bars represent the algorithms that are more accurate. The first bar is our optimized elastic net that has this prior knowledge, and the rest are standard predictive algorithms. On top, we have our original cohort, and on the bottom, we have our validation, our blinded validation cohort that the algorithm had never seen before. Originally, all these algorithms were doing really well, but on the bottom, when we look at our validation cohort, only the model that has this prior knowledge incorporated remains statistically significant after Bonferroni adjustment for multiple hypothesis testing. So this was published just a few months ago in Science Immunology. On the left, again, we have our training cohort, and on the right, we have the same model applied to postpartum samples after the dashed line. You can see that the immune system starts to gradually recover and get closer to a non-pregnant state a few weeks after delivery. We can go back to that correlation graph that Brice showed you and highlight the nodes that are particularly going up or going down during pregnancy. Larger nodes here represent nodes that on a single variate level by themselves are highly correlated with gestational age. And the smaller nodes are nodes that are not as correlated with gestational age. The colors represent their coefficients from our multivariate model. The red ones are highly associated positively with gestational age, and the blue ones are negatively associated. So Brice can now tell you a little bit about the biology of these findings and how they can guide future biological investigations.
Yeah, and what's interesting in this representation of these immune features overlaid on the overall immune networks is that we realize that they belong to various communities of features that can be characterized based on their most important attributes. And this can be a cell type or signaling pathways. And really what this is doing is it's pointing us into the direction of how the various immunological compartments are experiencing pregnancy. Now, the important communities were pointing us into the direction of NK cell changes, dendritic cell changes, and granulocyte changes, as well as adaptive responses, particularly in naive T cells. Now, reassuringly, some of these features are very consistent with what is known of the immune response to pregnancy, and others are pointing in the direction of interesting hypotheses. So I want to show you now the most important or most predictive features of the model. And that's important because it will guide future studies and the development of new mass cytometry panels in ongoing and future studies. The number one feature is in pink here, belongs to the community of features in pink. And it was pointing at this phosphorylation of the transcription factor STAT5 in naive CD4 T cell. And you see that it progressively increases during pregnancy to levels that are much higher than in the postpartum state. Now, as you may know, STAT5 plays multiple function in the differentiation of CD4 T cells, but downstream of IL-2, one of the most important role of STAT5 is to guide the differentiation of CD4 T cells to the peripheral Treg or to the Treg cell type, particularly the peripheral Treg cell subset. And so it promotes the differentiation of peripheral Tregs and their stability. Now that's obviously very important and very reassuring. We know from Alex Surdensky's lab and Sing-Sing Wei's lab how important Tregs are in the maintenance of fetal-maternal tolerance during pregnancy, and how from animal models, how this dysregulation of these cell types leads to premature birth and the loss of fetal-maternal tolerance. The other feature was pointing us in a totally different compartment of the immune system. It was pointing us to the direction of innate immune responses. And so here in the red dot in the orange community were all communities of immune features that were pointing at the toll-like receptor 4 response to LPS in innate immune cells. And the red dot is the feature in dendritic cells, myeloid dendritic cells. And what we're learning here is that the NF-kappa B response to LPS is dampened originally at the beginning of pregnancy and then progressively increases during pregnancy. And that's also interesting because we know mostly from animal models, from Sylvain Shemtov's group in the University of Montreal, that the response to TLR in innate immune cells in pregnancy, which is associated with the activation of the inflammasome pathway, is very much implicated in the maintenance of pregnancy. We also know from a recent paper by Romero's group that the inflammasome activation is the inflammasome pathway is activated at the onset of labor and is dysfunctional with the onset of premature labor.
All right, so moving on to the second part of the talk, what is special about this mass cytometry platform that we're using is that it's looking at individual cells. That's a very powerful thing to do because cells are the smallest functioning units of life, and you can actually figure out how to modulate them. It interacts with our microbiome, with what happens at a transcriptomics level, at a proteomics level, as well as the clinical context that these patients are in. And we wanted to understand the immune system in this larger picture. So in the same cohort, we produced a larger data set. We have cell-free RNA from the Quake lab at Stanford. We have a plasma proteomics assay that we produced together with Martin and Brice. We have metabolomics analysis of the same cohort by the Snyder lab, the mass cytometry data set that we just showed you, as well as fairly extensive microbiome profiling and analysis of plasma and serum, all done here at the Stanford. And of course, this is done in collaboration with a large bioinformatics team. Most importantly, Sajjad over there in a red shirt did much of this work that I will be talking about here. Our first goal was to understand if these data sets can interact with each other and make better predictions of gestational age. If you simply put all of these measurements into a correlation network, you're not going to see a whole lot simply because there are too many connections and too many correlations. So we used a similar, again, here Y is gestational age, X is the full data, and we try to do the exact same thing. We try to build an elastic net that predicts gestational age. First, we did that on one data set at a time. So each of these bars represent an elastic net built on each data set. Y-axis is again minus log 10 of p-value. You can see that some of these data sets are better correlated with gestational age than others. For example, on top, the microbiome data set doesn't move that much. We already knew that the microbiome is fairly stable throughout pregnancy. And on the bottom, the proteomics data set has a pretty good correlation with gestational age. We can look at that on our test set, and we will see that it generalizes fairly well to previously unseen samples. We can also apply that from samples towards the right of the graph. These are the samples throughout pregnancy that have been used for building a model, and then you can apply them to the postpartum samples. You can see that most of them start progressing towards a non-pregnant state, but some, for example, cell-free RNA has a higher quote-unquote memory level. It takes it longer to recover back to baseline. Now, if we take these models and combine them together and look at the final outcome, this is the correlation graph. The graph mostly consists of measurements from cell-free RNA and microbiome. This was a little bit surprising to us, but it turns out that that's happening because these data sets have a significantly larger number of measurements. They are completely overwhelming the predictive model. So we had to go back to our equations and add an extra term down here that would penalize the data sets that are producing a larger number of measurements. That helped improve our predictive power a little bit, as seen in the gray bar here, but that wasn't enough. Another issue was that some of the larger data sets may actually have less information in them. This is a large data set that only has 3 major clusters, maybe 4, and then this is a smaller data set that has many, many clusters. So from an information theory point of view, potentially it's measuring more biological processes and has more information content. We quantified that by using PCA. On the left, you have the number of measurements in each assay. On the right, we have the number of principal components that it takes to account for 90% variance. You can see that the mass cytometry assay, for example, has a very high level of modality, even though it doesn't have as many measurements compared to cell-free RNA or microbiome. So we had to modify our strategy to account for this. What we ended up doing is called a stack generalization. You basically build a model on each data set first, and then you take the predictions of gestational age from each model and use it as an input for your final predictions. You're stacking the predictions of each individual data set on top of each other, and then you build an additional model that makes a prediction based on all of those results combined. And that gives us this black bar here that is even more accurate than before and generalizes to test set. We can then ask if these models can be reduced to a limited number of features that can then be biologically investigated. Here we are starting on 200 features on the x-axis, and we gradually reduce them until we hit a point that we can no longer reduce them without losing our predictive power. And that gives us this correlation network of the features that are predictive from each data set. And we can then annotate the network using what we understand from the biology of these features. On the top, we have our microbiome data set. Here we have our proteomics data set. CSH1, CSH2 has already been reported in the literature to correlate with gestational age. It's right next to CSH1 at a transcriptomics level. We also have a number of metabolomics hits that we don't have time to go to, and our mass cytometry features are down here right next to the proteomics measurements. Brice can tell you more about this specific interaction.
So, as you noticed, the most predictive feature of our immune clock model, the phosphorylation of STAT5 in naive CD4 T cells, also appeared in the multi-omic integrated model that Nima just showed you, tightly associated with CHS1, a so-called human placental lactogen, which appeared in the proteomic data set as well in the cell-free RNA data set. So we wanted to evaluate the nature of this interaction a little bit further. Here is the proteomic data set that is represented again as a correlation network. And what we noticed was that CHS1 belonged to a cluster of proteomic features in red here that were all related functionally by the fact that they could activate the JAK-STAT signaling pathway. And particularly 3 of those were related to the JAK-STAT5 signaling pathway. And those included prolactin, CHS1, and IL-2. Prolactin and CHS1 can activate the JAK-STAT5 pathway through the prolactin receptor, and IL-2 downstream of the IL-2 receptor. However, when we stimulated whole blood samples from pregnant women at different times in pregnancy with these various ligands or combination of these ligands, what we noticed was that in our hands, only IL-2 could led to a robust phosphorylation of STAT5 in naive CD4 T cells. So now evidence of absence or absence of evidence is not evidence of absence, but perhaps what is happening is that the interaction between CHS1 and the STAT5 feature is indirectly mediated by the tight correlation that we're observing between the IL-2 concentration in plasma from women collected at the same time as we're doing the CyTOF data. So the IL-2 is increasing during pregnancy and tightly correlates with the STAT5 immune cell feature. And biologically, again, this is reasonable, and it's pointing at the IL-2 mediated differentiation of CD4 T cells into the Treg population.
For example, we know that Gardnerella plays an important role in some preterm births. We can go back to our data set and find Gardnerella in the microbiome data set, and then ask, can I use all these other data sets, the CyTOF data set, the proteomics data set, and so on, to find surrogate measurements for Gardnerella? You can basically do the exact same thing, except that you're excluding the microbiome data set now, and you're replacing gestational age with levels of Gardnerella in your predictive model. This is what we got when we tried to build a model on one data set at a time. What was really interesting here on both training and test set is that the data set that used to do really well is not doing as well, and the data sets that were not doing as well in the past are doing pretty well here. So it's important to not make general statements about one platform being better than another. It's just that one platform may be more relevant for one specific biological task. When we combine the data sets, again, the model that accounted for both modularity and size did a lot better than simply accounting for size without accounting for modularity. We can also look at the correlation network. Like before, we can reduce the model to the number of measurements that are necessary, and then we can put them in a correlation network and annotate the parts that we understand the biology of. We won't go into the details of this network. I will just point to the largest, most important feature from our mass cytometry panel, TCR gamma delta T cells, which are known to be important in mucosal immunity. So we are excited to see them, especially when we are looking at the interaction with microbiome and Gardnerella. Obviously something that we need to further investigate in the future. In terms of future directions for the bioinformatics part of the talk, obviously our small pilot study, we are very interested in looking at larger and more diverse data sets that have a significant number of preterm births. We're working with several other collaborators to expand our efforts into larger data sets from the developing world particularly. In terms of the bioinformatics analysis of these data sets, we are very interested in multi-objective optimization as well, especially for low resource settings. Here on the y-axis, again, we have minus log 10 of p-value. In the first bar, we have the full model that we've been talking about for gestational age. In the second bar, I'm excluding our most important data set, the orange one. And what you can see is that the model can retune itself to compensate for some of the lost predictor. So these other data sets can be combined in various ways to compensate for a missing data set. This helps us answer more complex questions in a resource poor setting. When we have to pick and choose between data sets, we can ask which combination of these data sets gives us the most predictive power, for example, per dollar. So we have cost and we have predictive power, or it can have even more objectives. It can have cost of feasible sampling from a particular site is and predictive power. So all these objectives in the future will need to be optimized at the same time. In terms of detecting deviation from normal, the statistics of it is not as trivial, especially when you have limited time points. That's another area that we're working on. We are also very interested in integrating these assays with population level information. Generally, this kind of multi-omics analysis is very expensive and it's not feasible to to assay an entire population. What we are trying to do is to approach this from the other end. Here we have a data set of 1.5 million births in California. Each dot represents a cluster of patients. You can see that there are specific clusters that have a higher black to white ratio. We generally know that African Americans have a higher preterm birth rate. So the idea here is to objectively select the samples from an analysis like this that would represent the entire population, and then go back and do a detailed multi-omics profiling on them. And then that would enable us to generalize the results to the entire population. And finally, our last interesting project is to integrate our findings with clinical phenotypes. Normally, here we are only looking at preterm birth versus no preterm birth, but there are more complicated phenotypes that we have access to. This is a correlation network of all the demographic information that we could get from our Stanford cohort. We can then use this correlation network as a backbone. I'm going to change its color to black. And there is the backbone down there. And then you can project your entire multi-omics biological data set on top of that clinical backbone to simultaneously understand, for example, the impact of stress on various biological modalities and on pregnancy itself.
All right, and then let me finish up with the rest of the future directions that are pertinent to the immunology side of things and the mass cytometry side of things. What we've shown you with the previous study is a model that predicted the gestational age during pregnancy. Obviously, the next question that's very important clinically is to determine whether we can predict the onset of labor or identify this immunological switch that may be associated with the transition from a non-laboring woman to a laboring woman in parturition. So we couldn't do this with a previous data set because although all these women delivered at term, some of them had C-section, some of them were induced, and so it wasn't a spontaneous onset of labor data set. And over the last year, two postdocs in the lab, Kaz and Xiaoyuan, have worked tremendously hard to recruit over 100 women that delivered between last February and this February. Of those, over 50 women delivered spontaneously at term. And so this creates a beautiful data set of samples that are collected during pregnancy, with an emphasis towards the end of pregnancy, towards the third trimester, and that will allow us to identify these immune events that perhaps predict the onset of labor. But of course, working on our work on term labor is part of our homework. Our major focus is to determine whether the immune clock of pregnancy is disrupted in pathological pregnancy. Is the immune clock accelerated, for example, in the context of preterm birth? And to do this, we have to expand our work and collaborate with other centers because the incidence of preterm birth is 10 to 12%. And so in order to validate our findings prospectively, we need access to larger cohorts of patients. I want to show you, before going into the details of these collaborations, I want to show you very preliminary data in the different contexts of pathological pregnancy, in the context of severe preeclampsia. This is exciting because it is perhaps speaking to the fact that with the approach that we took, we are oriented towards biological phenomenon that may be implicated in pathogenesis of abnormal pregnancies. So again, coming back to this feature of our immune clock model, the STAT5 feature, I want to show you the very preliminary data of a study that Xiaoyuan led, comparing two groups of women during pregnancy, a control a group that delivered normally at term, and then a group that suffered from severe preeclampsia. And you can see that in this very, very first pass of the analysis, just looking at this one immune feature, 10 out of 12 or 9 out of 11 control patients show that their STAT5 feature is increasing during pregnancy. Here we're comparing a blood sample at the second trimester to a blood sample drawn at the first trimester. So expectedly, we see this increase in STAT5 response during pregnancy in the control women. But virtually all but 2 of the women that suffer from severe preeclampsia have a completely different response, and their STAT5 response is going down during pregnancy. So now this is a univariate analysis where the data look fairly strong, and we're certainly expecting that with a multivariate elastic net model, we'll be able to separate very significantly these two groups of women. But finishing up on the idea of using a similar approach to understand the immunological abnormalities that may be associated or perhaps driving preterm labor, we were fortunate to be implicated in a large collaboration with the Amani Group. And this is a project that is funded by the Bill and Melinda Gates Foundation. The Amani Group is a large consortium of various sites in Bangladesh, Pakistan, and Tanzania. And they have an incredible capacity to recruit women during pregnancy and collect quality samples for various omics analyses. To give you an idea of this capacity, our collaboration started in early 2017, and in less than a year, those 3 different sites collected samples through pregnancy in over 500 women that delivered this year, of which 48 delivered preterm. So we're really hopeful that with this kind of collaboration, we'll be able to generate a large enough sample size to really be able to get to this question of immunological deviations associated with preterm birth and validate them prospectively. I want to finish up by showing you that we were, and this is not an easy task, but we're able to send samples back and forth. We send them smart tubes, and they send us a sample back to Stanford. And we started doing the QC of this data. And so far, so good. We're able to show that the samples can be stimulated, and we're getting the expected responses in these samples collected in this regard, in this example in Bangladesh. So these are very exciting prospects, and obviously all this work can be done without the hard work and intelligence of many students and postdocs in our labs. The top pictures are people that were implicated in the mass cytometry generating and analyzing the mass cytometry data set. So Amy Tsai, Ed Gagnon, Xiaoyuan, Kaz, and Diani. And then the bottom pictures are the people that are doing all the powerful analysis, bioinformatics analysis at Tanada, Sajjad, Tony, and then Martin Engs that I mentioned earlier in our presentation. And this is our acknowledgements slide. I want to finish by thanking our sponsors, the March of Dimes and the Gates Foundation, the CHRI at Stanford, as well as the Canadian Institute. And then, major collaborators in this project, obviously in the March of Dimes Center, where we are deeply involved in discussions of our work and other people's work. And this is led by David Stevenson and Gary Shaw and all the others here that are listed. Garry Nolan is always there to provide mentorship throughout our experience as junior faculties at Stanford. Rob Tibshirani and Trevor Hasty have been extremely helpful in the development of the ElasticNet algorithm that Nima developed, as well as our other collaborators for sharing their multi-omics data sets. Thank you.
Thank you. And I would point out that I did not develop the ElasticNet algorithm. We humbly use it.
Thank you, Dr. Gaudilliere and Dr. Aghaeepour, for that informative presentation. We will now start the live Q&A portion of our webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question drop-down box located in the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many questions as we have time for. Now let's get started. Our first question is, you use mass cytometry to define the immune clock of pregnancy in peripheral blood. What about the local immune mechanisms in the feto-maternal interface?
Well, thanks for asking this question. It's obviously a very important question. What we're doing with mass cytometry, as well as other omics platforms, is we're looking at the peripheral immune system or maternal blood in general. And so obviously the action, or if you want to say it another way, the epicenter of these various pacemakers of pregnancy is the fetus itself and the placenta. And so there are clearly really important mechanisms that are happening locally that likely drive these immunological response. And what we are measuring in the peripheral blood are signatures of these response. Whether they're causative or a consequence of these response, we don't know. And so in order to get to this question, we have recently engaged in a collaboration with the University of Chicago with Carol Ober. And she has started using mass cytometry to analyze the immune landscape, or the cellular landscape actually, of the placenta, using both at the maternal side and the fetal side of the placenta at the time of delivery. And in a study that we're doing together now, we're going to be able to look at the same time at the peripheral immune response as well as the placental immune response, and that will allow us to do an integrative analysis and see if there are meaningful correlations between the two data sets, and hopefully learn about the biology that is coming from these local mechanisms and have these ripple effect in the peripheral immune system.
Thank you. Our next question, mass cytometry dramatically increases the number of cell types and signaling pathways that can be measured. How do you control for false positives?
All right, I can take that one. When the number of measurements increase at the level that are increasing in the cytometry field, and especially with mass cytometry, the traditional statistical answer is that you have to increase your cohort size. When you have a larger cohort, or when you have independent cohorts, you can naturally control for false positives. But that is not always possible, obviously. So the alternatives are to use the sparse models that reduce the complexity of the models to very few measurements. When you have fewer measurements involved in the final predictive model, the chances of false positives also reduce. And finally, the thing that we are very excited about is the idea of incorporating background biological knowledge into the models, so that a sparsification process can be guided by our current understanding of the immune system. If there are specific features that we know are robust because there have been tens of articles already published about them, when we have a large number of measurements, those features should perhaps be prioritized if everything else is equal. So we are building on decades of immunological knowledge to populate our high dimensional space to account for that sparsity and for that lack of large number of observations.
Thank you. Our next question, what are the biggest considerations for incorporating mass cytometry data into future large-scale omics studies?
Mass cytometry is unique in that it increases the number of simultaneous measurements in various cell types, but it also doesn't produce an extremely high number of measurements like we would get from sequencing assays. So we have to balance out the number of measurements versus the amount of information that is provided. There are multiple ways to do that. We talked about one of them here. But generally, lower dimensional assays like Luminex tend to higher information content because they are measuring a specific biological processes that we understand very well. And as dimensionality increases, we can measure, for example, gene expressions of a large number of genes that all belong to the same pathway and are highly correlated. So mass cytometry falls somewhere between those two extremes. We have to make sure that the pipeline that we use does a good job at capturing that trade-off between number of measurements and information content. The second thing that I like to point out is that missing values are an important thing to deal with. Normally, in most assays, when you have missing values, it simply means that your assay has failed for that particular sample, or you just couldn't even collect the sample in the first place. But for cytometry, accounting for missing values is a little bit more nuanced because, for example, if you're looking at median of a specific signaling pathway in a cell type, and a patient happens to just not have that cell type, not have any cells in that cell type, what do you do with the median? Do you set it to 0? Do you use a predictive algorithm to interpolate the missing value? That's another more nuanced thing to think about when trying to integrate mass cytometry with other omics data sets.
Thank you. And it looks like we have time for one final question. What are the advantages of using a whole blood mass cytometry assay?
So, yeah, most of our studies are using whole blood, and I'll give you the advantages and also the drawbacks, and I'll start with the drawbacks. So, whole blood versus PBMC. Obviously, the whole blood system, the cells are fixed and banked fixed, and you can't revive them and analyze them as live cells as you would do with PBMC samples. That's a limitation. However, there are, in our hands, very important advantages. So number one, you have access to the entire immune system. Neutrophils in particular and all their subsets are accessible with whole blood assay, and you lose them with PBMC. Maybe more importantly, what we're in these exploratory studies that we are doing, where the biology is very much novel and hasn't been explored as much in humans as in maybe other diseases like cancer, you would say. It's really important for us to generate these initial data sets with a biology that is relevant to what is happening in vivo and is as close to endogenous changes, endogenous functional responses that are actually happening in the human blood. And with a whole blood assay, you have the ability to really fix these functional response within minutes of phlebotomy, within minutes of blood draws, and really be close to what is happening in vivo with minimal perturbation of your sample beyond stimulating for 15 minutes, for example. And then lastly, and this is very pertinent to the type of global collaborations that we have now, is the feasibility and the fact that the system that we're using, the SmartTube system, is very exportable. When we had to determine what assay to use, that we can standardize and replicate fairly, to the letter, the assay that we're using here at Stanford and replicate that in Bangladesh and Tanzania, for example. We wanted something that's very robust and easily implementable. And so here there's 4 simple steps. If we want to stimulate cells, as we do and as we're doing in Bangladesh, we can use tubes that have lyophilized stimulations. And so it's exactly the same process, whether it's the cells are stimulated or not. It's not extra burden. And all this is very much standardized. And I want to emphasize this part, that the ability to collect quality samples in many patients and in remote location is really paramount. And this is the quality of the data set in the end is something that sometimes is overlooked, and we think that with doing a whole blood assay we can really have this high quality, generate a data set of very high quality.
I would like to once again thank Dr. Brice, Gaudilliere, and Dr. Aghaeepour for their presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through June 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.